All right, so we're now going to talk about cellular reproduction. And this is kind of how you have cells from cells. So we're going to talk about how you can create new cells from existing cells. Okay, cellular reproduction is going to have to do with the birth of new organisms. So we will talk about both asexual and sexual reproduction. All right, and we'll talk about how this takes place. It's going to be different in asexual organisms than it is in sexual reproduction. And we are considered sexually reproducing organisms. This also has to do with just the production of new cells. So, all different parts of our body have cell turnover. So like our skin is going to need to be able to renew itself. Our different organs, some of them can have new cells that develop. And we're going to talk about this process of how this happens. Okay, so how we produce new cells from existing cells. Okay, we start off as a single-celled organism that is the fusion of egg and sperm, and we ultimately develop into an organism that is, trillions of cells, made of trillions of cells. Okay, now this process is going to have to do with cell division. Okay, and so this is basically a cell undergoing reproduction. Okay, so we're going to start with a single cell, and we're going to call this the parent cell. And then through the process of cell division, that cell is going to split into two identical new cells, and these are called daughter cells. Okay, so they are going to be genetically identical to the parent cell. Identical, which means that every single gene in the on the DNA is going to be identical. Okay, so this cell is basically going to split into two. We're going to talk about some things that have to happen for that to take place, but they are genetically identical to each other. Okay, before this happens, okay, so this is going to be an important step. You have to have the chromosomes or the DNA. So DNA has to be duplicated. Okay, so in order for you to start off with DNA in here, and we want to make two new cells with DNA in here, you have to actually double the amount of DNA so that when you get to the end product, each of these cells is going to be genetically identical to this parent cell, okay? So that means you have to make more DNA before that can happen, okay? So basically when they say duplicate, it just means copy. So copy of DNA made. All right, now when we talk about cell division, again, it can have to do with kind of allowing for reproduction. Okay, so asexual sexual reproduction, it can replace damaged or lost cells. Okay, so when you hurt yourself, you have to be able to make new cells to kind of fill in the gap of that. And it also permits growth. So when we start off as a zygote, we are a single cell. And then ultimately over, lots and lots of time, we're going to ultimately be a person with trillions of cells. Okay, so this is one cell. So the way that we go from one cell to trillions of cells is through the process of cell division. Okay, so cell division is going to allow for that. All right. So a little bit about reproduction, just briefly. So we're going to talk about asexual reproduction, and So asexual. Anytime you have the letter A or an in front of a word, it means kind of no. So this is not sexual reproduction. So this is asexual. So that there's no sperm or egg involved. There's no fertilization taking place. Okay, and this often is going to be happens in single-celled organisms, but not always. Oops. And so they're going to reproduce by dividing in half. Okay, and so when you have asexual reproduction, your offspring are genetically identical. Okay, so this is going to be kind of one type. We'll talk mostly about sexual reproduction in the end, because we're going to talk about the process of that. And asexual reproduction kind of takes place through this process called mitosis. And mitosis is also going to be how we just do normal cell division. Okay, so this is the type of cell division responsible for asexual reproduction and for growth and maintenance of multicellular organisms. Okay, so mitosis is what we're going to talk about first. We are going to talk about a different type of cell division, and so this has to do with sexual reproduction. Okay, and sexual reproduction is going to involve kind of an egg and a sperm. It's going to have two individuals that are kind of fusing their genetic material together. Okay, so it requires fertilization of an egg by a sperm. Okay, it involves production of gametes, and this is a new word that some of you may not know yet. And gametes are going to be eggs and sperm. Okay, so this involves a special type of cell division, okay, called, this is now a new one, meiosis. Okay, so spelled a little bit different from mitosis. This is going to be called meiosis. This happens only in certain locations, happens in basically the reproductive organs. Okay, and so those are going to be the ovaries and the testicles. Okay, so ovaries are going to make eggs, testicles are going to make sperm. All right, so just as kind of a review, mitosis is going to be for asexual reproduction, growth and maintenance. So we do mitosis all the time. When we need to make new skin cells, new liver cells, anything like that, it involves mitosis. Okay, so that's going to happen in us. Sexual reproduction, so this is going to be a specialized type. So this will be for sexual, kind of creating a new organism in a sexual way. This is called meiosis, and it happens only in the reproductive organs. Okay, and sometimes we call the reproductive organs gonads. Okay, and so the gonads then make gametes, which are going to be the egg of the sperm. Okay, and in humans, we have ovaries and we have the testicles. So we're now I'm going to show you a little bit of a introduction to what some of this kind of looks like. And all of these pictures are going to be in an additional PowerPoint that says kind of images for the lecture. So what I just want to point out is a little bit about cell division by mitosis. So, kidney cells are going to reproduce this way. So it would just be like one cell dividing into two. A human embryo is going to develop via cell division, so mitosis. So you start off again with one cell, ultimately going to be a trillion cells big. Certain organisms like amoebas, so single-celled organism, some multicellular organisms like sea stars, theirs is a little bit different. This is actually considered regeneration. So if you take off one of the legs of a sea star, the leg itself will start to grow a new body, and the other sea star will actually grow a new leg. And so the process is through mitosis. Okay, and then there are certain types of plants. Succulents are really good for this, but they will grow from clippings. So you basically are taking part of the plant, putting it and cultivating it, and it will grow from that. And so that is considered a kind of an asexual type of reproduction because there's no egg and sperm involved in that. All right, so we're now going to move on to talking about the DNA, because the DNA is going to be really important to this process. So we need to start talking about chromosomes. Okay, and so what chromosomes are they going to be kind of long DNA molecules. Okay, so it'll be like a full strand of DNA can be considered a chromosome. However, when we're kind of talking about chromosomes, we are not talking about just DNA. There are usually going to be other proteins involved. Okay, so just to back up a second. So long DNA molecules, so these are going to contain thousands of genes. Okay, so these are going to be what tell, what color hair do we have? What color eyes do we have? Everything that is going to encode for protein synthesis is going to have to do with these genes. Okay? Now, when you have the chromosomes, it kind of depends on what species you are for the number. So the number of chromosomes is going to depend on the species. And we're mainly going to talk about humans, so we'll talk about this. So humans have 23 types of chromosomes. Okay, so that means like we have chromosome 1, chromosome 2, chromosome 3, chromosome 4, chromosome 5, up until 22. We then have a little bit of a different situation. We'll talk about chromosome 23. But basically, humans have 23 chromosomes, and we're going to have 2 of each chromosome. Okay, so 2 copies of each chromosome. All right, and the copies, one copy is from mom, and one copy is from dad. Okay, so when you have a chromosome 21, chromosome 1, you're going to have one that's from your mom. So we could just say that this is mom's. And let me just get a different color and say, here's chromosome number 1 from your dad. Okay, so this is going to be dad. This is going to be mom. And so you're going to have, for chromosome 1, we have 2 copies. You have the one you got from your mom and the one you got from your dad. Okay? And so that's going to happen for each of the 23 chromosomes. You get one from mom, one from dad. Now, one way that we talk about chromosomes is we kind of use the letter N to represent kind of the number of chromosomes. Okay, and there are different organisms that we are going to see that some of them have just one copy of each chromosome, so we'll call that 1N. Other organisms have 2, and in humans, we actually have different stages where there's times where we have just 1 copy and other times where we have 2 copies. We normally have 2 copies, but what this is actually saying is saying 1 times the number of chromosomes. So for humans, 1 times the number of chromosomes is 23. Okay, so that'd be 23 chromosomes. This one, 2n, would be 2 times the number of chromosomes, which is 46. When we talk about 1n, this equals a haploid. This is just a new term. Oops, did I spell that right? No, it's OI. This is a haploid. That means just 1 copy of a chromosome. And when you have 2n, it's called diploid. Okay, so humans are going to be diploid organisms. So the majority of our cells are going to have 2 copies of all of the chromosomes. The only time that in humans, I'll just talk about this. This is most cells. In humans, we have some cells that are going to have just one copy, and these are going to be the gametes. And we'll come back to these later and talk about gametes. Okay? So this will mainly come into play later on when we're talking about meiosis, but you're going to see this in textbooks, and if you watch any other videos online, the number n stands for the chromosomes, and then you can have 1n, which means 1 set of chromosomes, or 2n, which means 2 copies of each chromosome. Okay, so a little more about the actual structure of chromosomes. So chromosomes are not just DNA. Okay, so a chromosome is actually made up of, just write this, chromosome. Okay, so is made of something called chromatin. Okay, and chromatin is DNA plus proteins. Okay, and so what this looks like is here is DNA, so we have the double helix, and then we have these proteins called histones, okay? And the histones are going to be kind of these little structures that the DNA is actually going to wrap around. So that is going to be the protein. So if I say DNA, protein, this is going to be histone. Okay, so there are proteins. So the DNA is going to be tightly wrapped around these histones, and then they're even tight, kind of wound around each other, and we ultimately form something called this nucleosome. I usually don't talk about nucleosomes too much because there's a lot of terminology when it comes to chromosomes. The main thing I want you to know is that chromatin is going to be DNA plus these histones, okay? The DNA then kind of even tightly wraps around itself over and over and over again until we get a structure that kind of looks like this, okay? And so I'm going to talk about this structure. And this is going to be kind of the standard way that I will draw the actual chromosomes. So here is what we kind of draw a single chromosome looking like. Okay, so it's not exactly what we see in, if I'm showing you right here, it'd actually just be half of this. So this would be the chromosome right there. Okay, so we need a little bit of information about the chromosome. So we can call this a chromosome. But I mentioned prior to this that you're always going to have kind of a copy from your mom and a copy from your dad. And so if we're talking about just one chromosome, I'm going to draw this. So we'll say that this is your mom's chromosome that you got from her. And you have this one. This is a chromosome you got from your dad. But they're the same chromosome. So they're going to be, we'll say this is chromosome number 1. Okay. And you would have chromosome number 2, 3, 4, and you'd always have one from mom and dad. So what we consider these 2 chromosomes together, this is going to be called homologous chromosomes. Okay, and so what that means is that these chromosomes, they are the same chromosome, with all the same genes, with the same genes. However, some of them could have different versions of genes because they're coming from two different individuals. Okay, so same genes, but possibly not the same version of a gene. Okay, and I'm just going to give you an example of this. So let's just say that your mom has brown eyes and your dad has blue eyes. So the gene for eye color is going to be found on the same chromosome. So if this is chromosome 1, we'll say this is where that eye color gene is going to be located, but the one you get from your dad will have blue and this one's going to be brown. So they're still the same gene, it's just a different version. It's like the blue versus the brown version of that eye color gene. So that is what we mean by homologous chromosomes, okay? Now, in this whole process, when we're starting to talk about mitosis and meiosis, we have to go through DNA replication. So we're gonna make more DNA. And so what's gonna happen is we're gonna make additional copies of the DNA. So I'm gonna start on a new piece of paper. Let me just redraw that, what I did for kind of mom and dad. So here's mom. And it kind of helps if you have two different colors so then you can actually tell. So that's dad. And so when we go through the process of DNA replication, because at the end we need to be able to make more chromosomes because you have to split them amongst two different cells, you are then going to make identical copies of each of the chromosomes. Okay, so we're gonna make another version of mom's in this blue, and we're gonna make another version of the dad's. Okay, and so I already told you that these two together are considered homologous chromosomes. However, what these two new things that we made, so kind of the blue and the orange, these are now called sister chromatids. Okay, so sister chromatids. Sister chromatids are identical. Identical copies of chromosome. Okay, so these are going to be identical to each other. So if we're talking about that eye color thing, so if we said dad had blue eyes, both of these are gonna have the blue-eyed version of the gene. Both of these are gonna have the brown-eyed version because they are identical copies. So when we're looking at this, we have homologous chromosomes, which are the same chromosome, but different version, could potentially have different versions of genes because one's from mom, one's from dad. When we make sister chromatids, these are identical copies of one of those homologous chromosomes. So here we have mom's DNA, identical copies to each other, so they're considered sister chromatids. These two orange ones from dad are called sister chromatids. You cannot consider an orange and a blue together a sister chromatid because they are not identical. Those are homologous chromosomes. Okay? So when we start going through the steps of mitosis, we're going to kind of discuss how we're going to separate the actual, in the first case, sister chromatids from each other. Because at the end of this, what we want to happen is we want each cell to end up with one copy of mom's DNA, one copy of dad's DNA. So we have to make these copies. Okay, so I'm gonna draw this a little bit different now so we can start talking about duplicating chromosomes. And I will talk about the actual process of DNA replication in detail in a later lecture, but for now, this is going to be kind of what we'll talk about. Okay, so this has to do with DNA replication, which DNA replication means making copies of chromosomes. Okay, so during this process, each chromosome is copied. Okay, and I'm gonna go back to what I always do. So we have our dad's chromosome, we have our mom's chromosome, Okay, and during the process of DNA replication, we are going to make a new blue one for moms, new orange one for dads, okay? Now, when we produce new DNA, we have they're actually bound together in the center, and we call this center part the centromere. Oops, centromere. Okay, so both of them would have that centromere. And I just want to make this again clear. So these are sister chromatids, as are these. Okay, so these are sister chromatids as well. All right, so during the process of cell division, what's going to happen is that we are going to kind of have our chromosomes line up, and I'm going to talk about this in more detail, but I just wanted to briefly show what this looks like. And then through the process of cell division, we're going to have kind of the orange sister chromatids separate from each other. The blue sister chromatids are going to separate each other. And we're going to end up eventually with kind of, this will be one cell, this will be one cell, okay? And they're going to have the correct number of chromosomes and the right types of chromosomes. So now we're going to talk about what's actually happening here. Okay, so for now, again, all you need to know about duplicating chromosomes is that you create a sister chromatid, which is an exact identical copy of each chromosome. Okay. And let me just show that one more time. So here's from the kind of textbook. You have a single chromosome. So we'll just say that this one is your mom's. This is from your mom's chromosome. This is mom's version. Then through the process of DNA replication, we have two of them, so they're sister chromatids. And then at the end, those two chromosomes are going to separate and go into each of their own cells. Okay. So we're now going to start talking about the actual process of mitosis. Mitosis is going to be part of this larger kind of process called the cell cycle. Okay, and what the cell cycle is, and I'm going to I'll show this to you and then I'll come back to it, is basically the lifetime of a cell from the point where it is created. So it's gone through, like, it's just been replicated, there's cell division has taken place, and so we have our cell, and it's going to go through this process until ultimately it is then going to divide itself. Okay, and so this is called the cell cycle. Okay, so it's an ordered sequence of events. Okay, and it's from the time a cell is first formed. Okay, so it's just been kind of divided and it's a new cell to the time where it's actually going to divide, to when it divides. Okay, and so you can kind of think of this as the lifetime of a cell. Okay, now the cell cycle is going to have different parts of it. The majority of the cell cycle is going to be called something called interphase. Interphase is going to be this entire part right here where you have from here to here. So this is all going to be interphase, and then we're going to have a very brief period of time that's going to be called mitosis, or the mitotic phase. Okay, so most of cell cycle is spent in interphase. And interphase is going to have a couple steps that are happening in it. So if you look back on this, there are these different stages of interphase, which we're going to kind of talk about. So let me just put interphase. Okay, and so what's happening during interphase is that the cell is basically doing its normal thing. So if this is a cardiac cell, or if this is a liver cell or a kidney cell, it's going to be doing what it needs to be doing, okay? So cell is carrying out routine activities. Okay? It also, during this time, is going to double everything in so during a specific period of time, it's going to double everything in the cytoplasm. It's also going to synthesize new DNA. Okay, so that DNA replication is going to happen. And when I say double everything in the cytoplasm, I should say organelles. Okay, because at the end of this, if you're going to have two cells, you need to have a mitochondria for each of the cells. Okay, and it's going to grow in size. So that is what's happening here, okay? And so we'll kind of define what's happening in each of these stages. So we have what we call G1, or first gap. We have the S phase, which is when DNA synthesis is actually taking place. So you can actually see how the chromosome is now duplicated. And then in G2, or second gap, we're going to have kind of growth taking place. All right. So the three subphases, so interphase. So three subphases. We're going to have G1, S phase, and G2. Okay, and they happen in that order. So the first stage is majorly going to be about growth. So the cell is going to get larger. The S phase is going to stand for synthesis, which means DNA replication. Okay, so this is the stage that new DNA is being made. There are lots of questions on practicals and lecture exams that have to do with this S phase. So when we ask you, when is DNA synthesized in the cell cycle, it is during S phase of interphase, okay? And then gap 2 is going to be kind of preparation for cell division. Okay, so we need to make all the new organelles. We need to make sure that there's going to be enough material for each cell to end up identical to the first parent cell. Now, what you're going to notice is that we also have this other little stage, which is going to be called the mitotic phase. And so this is a very small, short period of time in the entire life of a cell. Okay, but we're going to mainly focus on this stage right here because this is how the cells are actually going to divide. So the other part of the cell cycle is going to be called the mitotic phase. Okay, and when we discuss mitosis, mitosis only takes place during the mitotic phase. Interphase is not a part of the mitotic phase at all. Okay, so it is completely separate. It's preparing for mitosis, but it is not part of mitosis. Okay, in the mitotic phase, we're going to have two different processes taking place. We're going to have mitosis, which is going to be when the nucleus and all its contents, which is DNA, divide evenly into two cells. Okay, so that's all about the DNA. The second part is called cytokinesis. And this is about kind of the cell cytoplasm along with organelles is divided into two cells. Okay, we often talk about mitosis and cytokinesis kind of as one thing that's happening. Cytokinesis, this last part, is going to happen at the very end of what we call mitosis. And so I'll discuss more about cytokinesis in a second. But the whole purpose of these two stages is to kind of, when you combine mitosis and cytokinesis, you produce two genetically identical daughter cells. Okay, so that is going to be what's happening during the mitotic phase. So lots of stuff is happening during this phase. Okay. Now, before I start kind of actually going through the steps of mitosis, the main thing we want you to kind of get familiar with mitosis is what the stages look like in cells. But before we do that, we need to talk about what interphase looks like, because you have to be able to kind of pick out what it looks like before we actually have mitosis taking place, okay? So I'm going to be drawing these to the best of my ability. So when we have interphase, all of the DNA is going to be located inside the nucleus. So this is the nucleus. Okay, and we're going to have all the chromosomes are going to be basically decondensed. So they're not going to be in a very tightly packed form. Okay, so inside of this is going to be uncondensed chromosomes. Okay, so we're not really in that chromatin form at this point. I'm also going to draw two other little structures. Okay, we're not showing most of what's going on in the cell, just kind of the important parts. But these structures right here are called the centrosomes. And we've talked about these before, but these are organelles that produce microtubules. Okay, and we're going to call these spindle fibers, which are basically kind of very important in how the cell divides, okay? So they aid in actual cell division, and I'll kind of point them out to you in a second. So this is before mitosis. So interphase is not part of mitosis. When you look at a cell in interphase, you cannot see chromosomes in the nucleus. You can see a nucleus, but it's just kind of fuzzy inside. You often will not be able to see the centrosomes, but because we're drawing this out, and I'll show you some cartoons, you can actually see them in some cases. All right. So now we're going to move on to the actual stages of mitosis. So I'm going to write them out first, and then I'm going to draw these for you and show you what they look like in actual cells. So the first stage of mitosis is called prophase. Okay, then we're going to go through something called metaphase, anaphase, and the last stage is telophase. And we usually combine this with cytokinesis. Okay, so I'm going to start going through these different stages and talk about what's actually happening inside the cell during them. Okay, so for prophase, this is the first stage of mitosis. We are going to divide prophase into early prophase and late, okay? During the very beginning of prophase, what's going to happen is that the chromosomes condense. So you can actually start to see the chromosomes visibly inside of the nucleus. Okay, we also have our centrosomes, those little structures that are going to create the spindle fibers. They are going to start to separate. So they are usually right next to each other in the cell, and they're going to start moving apart. And spindle fibers start to form. During late prophase, we're going to have the nuclear envelope. Okay, so this is the structure that's keeping all the DNA inside of the nucleus. Nuclear envelope is going to break up, so it's basically dissolving. And we're going to have the spindle fibers attach to the centromere of the sister chromatids. And I will show you what this looks like. Okay, so this is what it looks like in an actual cell, and then I'm going to draw part of it for you. So here is early prophase, and so what we have right here is you can start to see the individual chromosomes. They eventually will break out of the nucleus, but this is what we kind of see in prophase. All of this kind of greenish yellow is going to be the spindle fibers and the centrosomes, okay? Now, during kind of later prophase, you can see that the centrosomes have moved, they've started to move apart from each other. The chromosomes are now free. They are no longer inside the nucleus. And they're actually, the spindle fibers, which are these green, are going to start to attach to the actual sister chromatids. And so I'm going to draw kind of a cell, very simplified cell where I'm not showing very many different chromosomes. Okay, but these are going to be sister chromatids because at this point, remember, after interphase, we already have our DNA has been replicated. So I'm kind of drawing them so that they're not right next to each other. Okay, and we have our centrosomes. And we're going to have these things called spindle fibers that are going to start to kind of attach. And so remember when I was talking about the sister chromatids, they are connected to each other at the centromere. And so what a spindle fiber is going to do is a spindle fiber will actually attach to each of the sister chromatids on the different sides at that centromere. Because eventually what's going to happen is these are going to pull apart from each other. So we can have the spindle fibers start to form, and they're, it's much more chaotic in an actual cell because there are lots of chromosomes. I'm only showing 3 sets of chromosomes right here. Each of these are going to be sister chromatids, remember? So they are identical. They're going to pull apart during mitosis. Okay, so this is going to be what prophase would look like. Now moving on, we're going to go into the next stage, which is called metaphase. And the main thing that happens during metaphase is that the chromosomes line up in the middle, in center of cell. Okay, so if we look at this, previously they were kind of off to the sides. Now what's going to happen is all of the chromosomes are going to line up in the middle. The spindle fibers are going to be attached to the sister chromatids. Okay, and so this would be metaphase. We still have those centrosomes over in the corners. Okay, this is what this actually looks like. It's very chaotic. You cannot tell that everything's lined up in the middle because there are a lot of chromosomes. But this is just kind of a very simplified version of what that would look like. So you'd have all of your chromosomes lined up in the middle and they would be ready, okay? Our next step is going to be anaphase. And during anaphase, what's going to happen is the spindle fibers pull apart sister chromatids. Okay, so in this one, what I'm going to kind of draw is that now this, the chromosomes, the sister chromatids, have now been pulled apart from each other and are being pulled to opposite sides of the cell. Okay, and our last little ones. Okay, so what's ultimately going to happen is we're kind of separating this structure, and we're going to start pulling these apart so that each cell is going to have the same number of chromosomes. Okay, this is what anaphase would look like, where you can start to see the separation of the chromosomes. So all these chromosomes are going to be headed over on this side of the cell. All of these chromosomes are going to be heading over here until we get to the very final part of the process of mitosis, which is going to be telophase. And I'm going to say / cytokinesis. Okay, and so what's going to happen in this is that the chromosomes are going to actually be kind of completely separated, and the chromosomes are going to start to uncoil, so they're going to not be in their chromosome tight form anymore. Uncoil. The nuclear envelope begins to form again around the chromosomes. We're going to have division of the cytoplasm. Okay, and what this is going to look like is we start to kind of see the separation of the cells. Okay, so all the chromosomes that were originally, attached, they're going to start having the nucleus form around them. And the chromosomes are actually going to start to decondense. So they ultimately are going to start to look like this, where you can't really see individual chromosomes anymore, just long strands of DNA. When we go through the process of cytokinesis and actual cell division, we have what kind of starts to form, which is called a cleavage furrow. Okay, and so ultimately this will complete and you'll have two cells that are going to separate. Sorry about that. So you're going to have one cell over here and one cell over here. They're going to each have the same number of chromosomes, all the different parts of the cell that they need. Now, what this kind of looks like is, here's the very last stage. So you kind of start to not see the individual chromosomes anymore. We're going to start to have a major division taking place. And so these will no longer be connected to each other. And we'll have the nucleus will form around the chromosomes. Okay. Now, when we're talking about plants, so this would be everything I've just talked about is in the animal cells. Plant cells do something very similar, but the very end is a little bit different because you have to remember that plant cells have a cell wall. And so when we have this last step where you start to have your kind of both your nuclei are forming and you have all your DNA in the different ones, what happens is there's these vesicles that have cell wall material and they start to spread themselves out in between the two sides of the cell, and ultimately as more comes, they will completely separate. Okay, but it's a little bit different. There's no cleavage furrow in this type. Okay, and so this is what an actual cell, this is the animal cell with the cleavage furrow. So the spindle fibers start to kind of pull the two cells apart. Okay, so we can have this contracting microfilaments. They will then kind of pinch these two apart, and then you'll end up with two different daughter cells. Okay, so we've kind of gone through the process. So major things that you need to know are going to be kind of what's happening during the different stages. Let me just write this. Okay, so need to know. So first of all, you need to know stages of interphase. Okay, and then the stages of mitosis. Okay, and what's happening in those. So you have to have G1, S phase, G2, and remember this is when DNA is being replicated. And then we have prophase, metaphase, anaphase, and telophase / cytokinesis. Okay, so make sure that you're able to identify those. with images and also kind of the different parts of it. Now, when we talk about the cell cycle, we have to bring up cancer, because cancer is going to be a disease of the cell cycle. So we're going to briefly talk about cancer. Okay, and so cancer cells. And what's happening is this whole process of mitosis is out of control. So it's dividing out of control. So what happens is, in normal situations, the cell cycle and cell division are tightly regulated. Okay, so there are lots of checks and stops and go-ahead signals that have to take place. Okay, and so we call this the cell cycle control system. Okay, there are different enzymes and proteins that are involved in this that send kind of stop and go ahead signals at specific points in the cell cycle. So basically what's happening is there are constantly, these different mechanisms are in place to check to make sure everything is happening in the way that it should, okay? And so what happens with cancer is that something goes wrong with the kind of the checks and balances of this. So cancer is a disease of the cell cycle. So when the cell cycle starts to kind of go haywire. So cancer cells do not respond normally to cell cycle control. Basically what happens is they start to divide excessively. Okay, and when they do this, they can invade other organs and tissues. Causing problems. Okay, now how cancer usually starts to develop is that there is going to be a cell that undergoes a kind of genetic change, which we are going to call a mutation. And usually that mutation is going to be in the cell cycle control system. So some gene that's involved in the cell cycle control system is going to be mutated and changed. Okay, so in cell cycle control system. So what this means is that the cell cycle control system is no longer functioning. So it's a control system no longer functioning correctly. Okay? And there are times in our bodies that cells can get kind of out of control in their growth. And depending on how and what takes place in that process, sometimes you can have things called benign tumors, which are just going to be growth of cells, but they stay all in one location. Other times we're going to have things called malignant tumors, which are going to start to spread into other areas. Okay, and there's lots of different types of treatment for these different types of cancers, including surgery, radiation therapy, and chemotherapy. There's also new types of medications that kind of look directly at the type of cancer and try to track how it evolves, and is very personalized in trying to combat it. But the big thing is, the reason why we talk about cancer here is because it all has to do with mitosis, okay? Mitosis kind of going out of control.